Hello, today we'll be talking about a microscopic look into the cell cycle with onion root tips. Our team members are
Nernar Nakhaleh.
Alexander Lay.
Kaitlyn Malacara.
and Haya Darkzali. So here's the agenda. First, we have the title slide, which we just went over, the background and hypothesis, designs and methods, timeline, safety and equipment, and the predicted outcomes for this lab.
All right. Hello, I'm Kaitlyn, and I'll be presenting our background and hypothesis. So jumping straight into it, we're going to be discussing the background. For this experiment that we are altering, we are looking straight at the Lab 6 mitosis and meiosis from our fall 2024 semester's course. In this specific lab, we looked at, for procedure 2 of this lab, we looked straight at an onion root tip cell and counted out 50 different cells that were going through different stages of mitosis. And once we recorded all of those, we got to calculate out which one was the most commonly seen during that particular life stage. While doing this with our 50 cells, we determined that prophase was the most common one that we were able to observe. But for our alteration of this lab, we're going to be counting out 100 of the cells instead of only 50. So for our hypothesis, which is we've decided to settle on the statement of if we counted 100 onion root tip cells instead of the 50, then the data collected will be slightly different from the experiment originally counting only 50 cells, because there will be a wider range of cells in the different stages of mitosis that will shift the ratios of the most common stages. So as discussed before, this is kind of because when we looked at the only 50 cells being counted, we've noticed that prophase was the most common, but with this new doubled data sample size, we don't know, will that be the same? Will that shift? And so that is exactly like what we're trying to determine with this specific experiment and what we are demonstrating.
Hi guys. Okay, so basically now we're gonna go ahead and jump into the explaining why we ended up adding things to the experiment. So there are two different things that we decided to just add in there as precautions. One of them as precautions, the other one was absolutely needed. The first one being the as precaution one is in the case that they decide to give us an onion root tip, which does not have any roots that are sprouting. And so we cannot really start our experiment due to that factor. What we're going to go ahead and do, since the lab is going to last for approximately 2 days, we will take the onion and soak it into a beak of water, and that what's going to happen is it's going to hopefully stimulate enough growth that we're able to shave off those cells and then proceed with our procedure as normal, or as experiment as normal. In the case, which we highly doubt this will happen, by the way, but in the case it doesn't happen, we will just carry on with our experiment as normal. And yeah, that's the first precaution. The second one being, which is not a precaution, it's something that we will absolutely have to do, is the ethanol acetate, which is basically the reason that we're going to go ahead and use ethanol acetate is it's cheaper to use and it's also safer to use, which is crucial for our lab. But that is the only two things that I really wanted to mention in this slide. Okay, why is this subject interesting as a whole? Why did we choose this lab to alter for our experiment? The reason for this is this lab, we all collectively found it very fascinating, because the idea that we can physically look underneath the microscope and see the process that is so common, it happens every single day, multiple times a day for continuously, it doesn't even really stop, but we still didn't fully understand it as humans. So the fact that we're able to contribute to that data, we just found it really fascinating. And honestly, that's why we really went ahead and chose this lab. All right, now I'm going to take you through the experimental designs and methods. So this picture over here is actually a picture that we took during one of our labs while looking at an onion root tip underneath the microscope. So we dyed it, as you can see, because otherwise we will not be able to see its structures. And then we took a look at it, and in this picture we can see the interphase, metaphase, anaphase, all of the really good phases that are out there. So I found that really fascinating. So we decided to add that picture that Haya went ahead and took for us during one of our labs. All right, so for the experimental methods, we're going to start off by getting an onion root tip, and we're going to shave a piece off. This piece should be approximately 1 to 2 centimeters. It should be as thin as possible. The thinner, the better. I also want you to make sure that the onion root tip is at room temperature. That is really crucial. The next thing that we're going to go ahead and do is we're going to place the onion root tip sample into a test tube, which has 2 mLs of 3% ethanol acetate solution. And the way that we're going to add the acetate solution is using a pipette. We're going to leave this for approximately 15 minutes. The reason we are going to go ahead and do this is because ethanol acetate, what it does is that it's basically stops the cell from proceeding to other phases. Why? Because it kills it. So it kills the cells, hence why the cells physically kind of like stop continuing on in the phases, which gives us a more accurate read because it doesn't deviate from anything. Sorry, I just got a phone call. I don't know if you heard that, but we're gonna go ahead and move on to the next phase. So after we left that onion root tip in for 15 minutes, what we're going to go ahead and do is we're going to pull out the sample and we're going to place it on a clean cover slip using tweezers, of course, not with our fingers. Haya also is going to mention that throughout this lab, it's crucial that we use tweezers. That is very important. We are going now to take out the sample and we're going to stain it. Why? Because if we don't stain it, it will be clear and it won't be like the picture that you are seeing currently on this slide. What we're going to go ahead and do, the way that we're going to stain it is by using a pipette. We're going to add a drop of 1% methylene blue, and we're just going to wait for roughly 10 to 15 minutes just for the dye to develop and make sure that it's all in there. And then we're going to go ahead and take our sample and we're going to place it underneath the microscope using the 400x magnification, and we're going to take a look at it, and then we're going to start classifying and choosing 100 cells at random and classifying them into whether they are in interphase, prophase, metaphase, etc. All right. Then comes the data collection and analysis. What we're going to go ahead and do is we're going to place all of the numbers for interphase, prophase, metaphase, anaphase, telophase, and then we're just going to write them all down in the first 2 columns. The second column, what we're going to go ahead and do is we're going to calculate the percentage of each cell in each stage. How we're going to do that is basically we're going to take the total number of, let's say, cells in interphase that we have found at random, again, from the 100 cells. So we're going to take that and then we're going to divide it by the total number of cells that we've counted, which is 100 cells. And then we're going to multiply it by 100. And then afterwards, we're going to take that percentage and we're going to multiply by 1,440 minutes. The reason we're going to do that is because a cell takes approximately 24 hours, or an onion root tip cell takes approximately 24 hours to go through all the cell cycles phases, which is why we decided to multiply it by the 1,440 minutes. And then, of course, there's 2 questions at the bottom which we need to complete. Afterwards, what we're going to take is we're going to take all of our findings and we're going to place them into a bar graph. The reason we're not doing logarithmic is just because we all decided collectively as students, because we are going to be conducting this in the future, hopefully, that we prefer a bar graph just for this experiment in particular. It's easier for us to read, so we went ahead and did that. The x-axis should be your independent variable, and that should be labeled the phases of the cell cycle in the onion root tip. So that would be the interphase, prophase, metaphase, anaphase, and telophase. And then I also mentioned that in the case you don't have a color coordination, kind of like this lab, you're going to assign interface to 0, prophase to 1, metaphase to 2, etc., and so on. The y-axis is going to be our dependent, and that is going to be the calculated percentage of cells in each phase. And then the control, obviously, for the experiment is that all of the cells are going to be conducted or going to be taken or counted from the same sample. We're not going to use different samples. We're just going to control that one for our experiment. We're not really going to use that information when conducting the bar graph, but we just wanted it to be in your minds. That is all. Thank y'all.
Okay, now I will be talking about the timeline, equipment, and safety measures for this certain lab we're doing. Now, the timeline is going to be a very much like summarized portion of what Nini was mentioning. She went into further detail, but the timeline again, so we're going to be reading this from left to right, from beginning to end, and this lab will take students only 1 day to complete. It will not take the whole 2 days because this lab is very simple, and it is something that students are able to do in one complete lab day. So we're beginning with the cutting the onion root tip in thin layers. Now here I actually have an onion with me with grown out tips. So we'll just be like cutting. This is minimum growth, but we'll be cutting from here. And then we're going to be soaking that in ethanol acetate solution for 15 minutes. Then we'll be transporting it with a tweezer on a clean slide. Students will dye the onion with methylene blue dye. This allows us to be able to see the cells more clearly. Obviously, if we don't have any dye, it's just going to be clear and we're not going to be seeing any cells in their growth. Now, obviously, what we're going to be seeing it with is the microscope, and we will be setting the microscope to 400x and examining the different stages. Again, for this lab, we will be moving the number up to 100 in total. And then after that, the students will complete the graph and the tables that are given, which will be shown in the next slide right here. The bar graph was made by me, and then the table was made by Nernar. And this is what students will fill out at the end of the lab for gathering data. Now, here are some equipment that is needed. So it's going to be divided into two parts. It's going to be the equipment provided and the equipment that is going to be needed by the Department of Bio 1201. Now, what is already provided is the dropper, the pipette. We already have that in the lab. The methylene blue dye, again, to stain the cells, the microscope and the microscopic slide so we can see the cells, a beak of water if just in case the onions does not have a full-grown root tip and we do need to have it grow. We'll set that in for 24 hours, and tweezers for departing the onions onto the microscopic slide. Now, what is going to be needed, and we'll ask the department to obtain, is an onion with an onion bulb, a razor blade for cutting the onion, ethanol acid, just in case that the department does not have much of it, and a timer. Now, a timer is something students can easily access. You can be using it through your phone. You can be using it through, the clock on the in the class, but that is not something that is we're heavily worried about for this project. Now, again, for cleaning up, we just want to make sure to clean and leave the table as it was when you first came. So, you come in, everything's organized, you leave, and everything is organized. Now, here are some safety and considerations. Now, every lab, the number one priority is safety for sure. That's the first thing we learn in the bio 1, and probably going to be the first thing we learn in bio 2. Now, personal protective equipment is key. Two main ones that we'll be focusing on is going to be gloves. Here's some gloves. Now, these are important to ensure we do not contaminate anything while using the ethanol acetate solution, and an extra layer of protection when we're going to be using the razor. So, making sure we don't burn ourselves, making sure we don't cut ourselves. Eye shields. Now, eye shields are highly recommended due to the fact that onions produce a chemical that irritate the eyes, the lacrimal glands that make tears in the eyes. I know a lot of people who are very sensitive to the onions and like they just end up like tearing up and they can't see anything. So eye shields do help with blocking out those chemicals from entering. Now, if the individual's hair is past their shoulder at length, please make sure to tie it up or clip it away to ensure lab safety. You know, we don't want anything in our hair. We don't want to leave with something in our hair and we don't end up noticing or contaminating anything. And then any jewelry that is worn must be taken off, including earrings, just to maximize the safety of this lab. You know, having rings on can lead to it being too sharp and cut the gloves, which also exposes us to things. And if for any reason the razor was handled wrong, please make sure to wash the cut or wound with warm water and soap and apply a first aid kit that is or should be in the classroom, and please notify the instructor. Thank you.
All right, now I will be outlining some of the potential outcomes that could come from this experiment. So due to the mimetic or likeness of the procedure to the host experiment, there's not going to be a whole lot of outcomes to consider, or in the very least, none that are too outlandish or radical. As far as I see it, there are three possible scenarios. There's the null outcome, the revelation scenario, or the chaos scenario. Now, the null outcome is simply this. Absolutely nothing changes and absolutely nothing happens. The increased sample size of the onion root tip cells will have zero bearing on the outcome of the reproductive cycle or the stage that the cells are observed in. And so this is the most likely scenario, as it's simply just a matter of statistics. Now, average statistics dictates that regardless of sample size, if a sample size is truly randomly sampled and it is representative of the sample population, then the results must and will be accurate and representative of the total population. To put it in an analogy or like situation, if you were to take a few thousand people out of a certain community of, let's say, total population of 2 million, but they were randomly sampled and deemed representative of the total population, then their trends would be likely to reflect the trends of the entire community or the entire population. This is why the null outcome is the most likely scenario, because the original experiment, statistically speaking, already took this into account. Now, in this scenario, we would observe that the most frequent and therefore the longest cell phase would still be prophase. Now, in outcome 2, this is the revelation scenario. In this outcome, the larger sample size would hypothetically allow us to have an increased look on the activity of onion root tip cells. This would allow for a very slight possibility that the generalized outlook of cell activity has either been overlooked, misconstrued, generally just wrong, and that on average, a different cell stage is the one that actually takes the most precedent. Let's say rather than prophase being the dominant longest taking, instead it's actually metaphase. Now, this would be a great revelation to the scientific community and ultimately would require further study to be verified. Now, in this scenario, we would find a new dominant cell stage in the cellular life cycle of an onion root tip cell, and as logic would dictate, that would be the cell with the highest frequency or the one that on average is spent the most time on in order to complete. This would add another cell stage to be the contender of prophase's #1 spot. Now, there's also outcome 3, the chaos scenario, as I called it, or the equilibrium scenario. To be frank, this is the least likely of the 3 scenarios. It's very low statistical probability and generally makes little to no sense, but it's still a possibility, and so I'm still going to outline it for the sake of argument. Now, in this scenario, rather than seeing one cellular phase rise above the others, due to the increased sample size, as mentioned before, we're allowed to get a deeper look and an increased look. And instead, we see, rather than prophase as we expect, all of the cellular stages having equal frequency and thus equal balance, and there would be no trend. This would certainly be chaotic, as we have established that each stage of mitosis takes a different amount of time to complete, and this would, of course, be challenged by our findings, as if this were the case, it would be very unlikely for all of the cellular stages to have an equal amount of time to complete, which is why I dubbed this scenario the chaos scenario, not necessarily by the activity in the cell, but more the activity that would ensue afterwards in our own minds and in the scientific community.
Okay, here are the references, and then a different page for the references, mainly for pictures, and then a thank you slide for watching this whole video, and some credits to SlidesGo for letting us use their templates.